Viraj. I am your biology teacher. So students, we have completed functions along with properties of plasma membrane. Right? You all have understood everything. I hope you all have noted down all the points which I have given. You see the blackboard is written, right? Okay. And as per our schedule, today we will start movement of water. In this class, you will be knowing how water particles or substances move through our cell in a human body or in a animal body. Okay. And I hope you all are doing well in your holidays and this pandemic situation. Please take care of yourself and read well. Okay. So we will discuss about activity number 2. What we will discuss, students? We will discuss about activity number 2. As I have always explained you all, while writing an activity, we must have our aim and materials required. So we need to go through both of them. So activity number 2, I'll write it here. Right, and along with that, I'll write aim of the experiment. What is the aim of our experiment? To study the movement of water in animals by using X. What? To study the movement of water in animals by using X. So by studying X, we will see how the materials are moving, how water is moving in and out of the cell. First thing. Second, what we need to write materials required. What materials we need for this particular experiment. So again we write what are the materials we need for this particular experiment. So 3 beakers we need. How many beakers, my dears? 3 beakers we need. And what we need? We need dilute HCl. We need dilute HCl. Along with that, we need salt. We need eggs. Because the experiment itself speaks, we will use eggs and we will study, right? Yes, we will use eggs. And what? We will use scale of measurement and tablespoon. We will use scale of measurement along with that we need spoon. Okay, we have now all the materials. Whatever we need, we have all the materials. Now we will perform our activity. Here we need to write initial activity, okay, and initial observation, then final activity and final observation. Okay, what is our initial activity? We will put both of the eggs, okay, in the dilute HCl and keep it for 4 hours. See what is it? It's initial activity. In the meanwhile, you can please note it down. Okay, here we will take the container. Okay, and we will put what? Both the eggs. Okay, here we will place dilute HCl, means this one is dilute HCl. We'll put dilute HCl and we'll place X and we will keep it for 4 to 5 hours. This is our initial activity. What we will write, we will take the beaker, we will fill it with dilute hydrochloric acid, we will keep 2X and we will leave this particular beaker for 4 to 5 hours. After leaving for 4 to 5 hours, what we will observe? That will be known as initial observation. That is known as initial observation. We will see what is initial observation, what we will get from this observation. We will get that. We will take it out. Now what we will do after keeping 4 and 5 hours, see, after this initial observation will come. Till now initial observation, it didn't come. Okay, function is still in, activity is still there. So we will place it, we will keep it for 4 to 5 hours and after keeping we will take them out. We will wash them. We will wash them. And we will wipe it with the cloth. We will wipe it with the cloth. Now we will observe. We will observe the changes and measure the circumstances of the cells dissolved because its made up of CaCO3. What we will observe, my dear? What is the changes we observe? We will measure. We will measure both the ends. This end goes like that. Now we will measure the size of both the eggs. When we are measuring, we have observed that both of the size was equal, but upper cell has been removed. Why? Because the cell is made up of calcium carbonate. The cell is made up of calcium carbonate, and calcium carbonate will react with dilute hydrochloric acid. It completely washed it, and we will have only egg, the fluffy egg. Okay, means ball-like, rubber ball-like it will be. Now, you see both of their circumstances are same. Okay. Now what we will do? We will perform final activity. It is done. Now we will go for final activity. In final activity, what we will do? In final activity, what we will do? We will take more 2 beakers. In 1 beaker, we will fill with water only, and we will place our egg. Okay. Other beaker, we will fill it with salt water. We will fill it with salt water, and again here also we will keep our egg. Now we will leave this for again 4 to 5 hours. Okay. Now take them out, wipe them with cloth. Now again measure their width, their size. Let us measure. We will see that now it is final observation. Now it is final observation. Final observation, what we will see? We will see that the size of the egg which is kept in the water has increased. Okay. Whereas the size of the water which is kept in salt water has decreased. It's swollen, it's shrunk. Okay, it is swollen. Its size when we are measured, it is swollen, but its size is shrunk. Okay, why? Why this thing happened? Why this thing is happening, students? Now comes learning part. Last what will come? Learning part. In learning part, we will tell that the egg is swelling because of osmosis. The egg is swelling because of osmosis. Let me write here. What I write? Here I write learning. And what I write here? The egg is swelling because of osmosis, right? Because of osmosis the egg is swelling. How? You have kept the egg inside the water, then what will happen? The water will come inside the egg and the egg will slowly swell because the outer layer of the egg is still acting as a plasma membrane, and the plasma membrane will allow water molecules to pass through it, right? So when water molecule is coming through it, it is said to be what? Swollen. And this kind of solution means this kind of solution where you are keeping an object and the object is swelling because of the surrounding, that is known as hypotonic solution. Hypotonic okay, what you will tell? Hypotonic solution. If you are keeping an object in a hypotonic solution, that object will be swollen. And now, suppose if you are keeping an object in a solution where the size doesn't change, then you will say that is isotonic solution. Now again, if you are keeping an object inside a solution where the size shrunk, that solution is said to be hypotonic. Hypotonic, hypotonic, there is a big difference. Hypotonic size increases, hypotonic size decreases. Okay, now when we kept inside water, what happened? The size increased. Okay, that is a kind of hypotonic solution. But now in second case what happened? The size shrunk. So what we have learned? Egg shrinks because salt solution is more concentrated. Egg shrinks because salt solution is more concentrated. Is that clear? Yes. Salt solution is more what? Concentrated. Okay. Now next thing what we have learned? Next thing we have learned is water comes into egg and osmosis. Water goes out from the egg is called exoosmosis. Okay, see, we will discuss what exactly it is. Here what happened? Water is coming inside. Here what happened? Egg is going inside. This will be known as osmosis, endoosmosis. Will be called as what? Endoosmosis. And this will be called as exoosmosis. Endoosmosis is called as exoosmosis. Is it clear? Yes. Okay. So this is what our activity number 2, and from here you have learned a lot of things. Okay. I hope you will keep up and you will note down the entire experiment. Okay, as it is written over there, and we will take a leave. And in our upcoming classes, we will be learning preparing of semi-permeable membrane as well as importance of osmosis in living organisms. Along with that, we will perform activity number 3. Until then, bye.